All right, thank you for watching this. This is a presentation about Salix recovery following Tamarix removal in riparian systems of the Western U.S. My name is Alex Goetz, and before I get started, I just want to mention that my co-author Ian Moffat will be available on the Q&A session as well to answer questions. So just to give some really brief background, I'm sure everyone is pretty much familiar with this dynamic, but the Southwestern willow flycatcher is a federally listed endangered bird that uses Tamarix often as nesting habitat because its native habitat is no longer present in a lot of these areas, and defoliation by the Diarrhabda beetle therefore may reduce these flycatchers' reproductive success because it is reducing its nesting habitat. So our research questions today are, first of all, whether it's possible to increase Salix cover following Tamarix removal in such a way that we don't see a net loss of flycatcher habitat. Second, whether there are particular restoration methods associated with increased Salix cover following Tamarix removal. And also whether there are particular environmental variables that are associated with increased Salix cover following Tamarix removal. So our methods for this, we used a large data set from riparian restoration projects across the Southwest. We had 243 treatment sites, 93 desirable reference sites with mostly native vegetation, and 79 undesirable reference sites with mostly invasive vegetation. And in addition to plant cover data, we also had data on treatment methods and abiotic conditions at each site. And this data came from a large data set that the lab has been doing a lot of work on in past years. And so I'll talk more about this later, but we conducted separate analyses on all Salix species as well as just Salix exigua. So our study area encompassed quite a bit of the Southwest. You can see it's clustered mostly around 3 watersheds, and you can also see the breakdown of reference versus removal sites within each of those watersheds. And I want to point out especially that this data includes sites from the Dolores River Restoration Project, which was in partnership with the BLM and Rivers Edge West. So for our data analysis, we constructed general linear mixed models that incorporated removal variables, biotic variables, and abiotic variables, so things like climate, water, roads, etc. And the way we constructed those models was using stepwise selection, starting with a lot of variables, and then after we had our significant variables, we conducted separate univariate analyses to show what was going on there. And we also removed 0 change rows for all of this. So if there was no Salix prior or after removal, we did not include those. And to talk a little bit more about our removal variables, so we had site type, first of all, which included sort of the primary type of removal that was done at that site. And then we also incorporated undesirable and desirable references there. So that sort of included, each of these sites had one kind of primary type of restoration work that was done to remove Tamarix. And then we also just had our reference sites in that same variable category. And we also looked at whether any herbicide was used at any point, and also whether there was active plant introduction done, and that includes planting and seeding. It is important to note that we don't have data on what exactly was planted, so it wasn't necessarily Salix, but we included it in the models. And then our dependent variables, so we did also look at final year cover. We're not going to be talking about that today. We're just going to be looking at cover change over time. And then, so we separately looked at combined cover of all Salix species and then also pulled out just cover of Salix exigua. The reason for that was that even though the flycatcher can nest in any of these species of Salix, exigua is the dominant Salix species in these sites. It was present in 83% of them, and it's also functionally distinct from many other species. It can reproduce vegetatively, so we wanted to consider it on its own, and then also see if we, would see the same patterns or different patterns when we look at that one separately from all of the Salix species put together. So the results of our model selections, this is what the output kind of looked like. So we were interested in looking at adjusted R squared of these to see like how well these models are predicting the variation in Salix cover at the end, or change in Salix cover rather. So we looked at marginal R squared because that is what just is looking at the fixed effects, so the variables we were actually interested in. We did have random effects as well, but we weren't interested in those. And so you can see here that this particular starting conditions led to 5 different models. And so this was just looking at Salix exigua cover change. And so we had 5 different models, partially because our climate variables were intercorrelated, and so we had sort of these factorial designs where we tried to just put one in each to start with. So we ended up seeing that herbicide use was important in a lot of these, site type was important in one of them, and then soil electrical conductivity and Salix exigua cover prior to treatment were both significant in all of those models. And then again, climate was pretty much broadly important, and we had a different climate variable in each model because they were intercorrelated, but those came out pretty frequently. And in general, just to really briefly summarize what we found with the final year cover, site type was pretty important in most of those. But then when we look at the change over time models, it really didn't come out very often. This was actually the only time when it came out in any of our change over time models. And so that kind of shows us that, you know, you can have an ending point that is going to be largely based on your starting point, but the change is not necessarily as affected by how good the site was to begin with. So just to talk about a few results that were interesting from this. So we found that soil properties were very important. So looking at Salix exigua on its own, soil electrical conductivity came out as a significantly negative relationship. Looking at all the Salix species together, percent sand was a significant positive relationship. Again, both of these are in the mixed model, not necessarily significant on their own. And so we saw that you know, saltier soils are generally bad, sandier soils are better, and that makes sense with what we know about the niches that Tamarix and Salix can occupy. And it also shows us that even after removal of Tamarix, the salt will stick around, and that is going to have a negative impact. So it's going to make it harder for Salix to regenerate even after the Tamarix is gone. The next result we found was that herbicide use generally has a pretty positive impact on Salix cover change over time, and you can see there is a lot of variation. Part of that is because there was a significant interaction with climate. So generally when there was more precipitation, you got kind of a better benefit from the herbicide use. If there wasn't enough precipitation, then it didn't matter as much. But yeah, both of these were significant. And again, just to reiterate, this was any herbicide used at any time. So also we found that existing Salix exigua cover has a negative relationship with change in Salix exigua cover. So if you already had a lot, it's not really going to be possible to get much more. So it seems like it exists in kind of a small niche that can be filled, and after a while that intraspecific competition becomes more important than any kind of, you know, increased reproductive potential that you would have had by already having some there. So when we looked at just removal methods in general to see if there were broad differences, but there really weren't any individual primary methods of Tamarix removal that were associated with higher cover of overall Salix or Salix exigua. However, when we looked at just cut stump sites, we do see a proportional negative relationship between Tamarix and Salix cover change. So as you lose Tamarix, you are more likely to gain Salix. Unfortunately, that's also vice versa, and we see both. And there is a lot of variation in this. A lot of them are clustered around this 0 mark with not very large gains in Tamarix, but in some cases we can see that you can get a lot of Salix without really losing much Tamarix, and you can also lose Salix without changing anything about the Tamarix. So we saw this trend overall, but it's important to note that, you know, there's a lot of variation and it's not a clean relationship. And then when we look at overall with all of the removal method types, we don't see that same proportional relationship at all. It was not significant. And so when we look at planting methods, we didn't see a significant effect of planting or seeding. There was a lot of variation, and part of that, again, is probably just because we don't know exactly what was planted or seeded in each site. And in fact, if we had something other than Salix planted, then we would expect to see less Salix just due to competition. So to circle back to our questions, first of all, is it possible to increase Salix cover in such a way that we don't have a net loss? So, you know, we did see that proportional increase after cut stump removal, but it did not compensate for the loss in Tamarix cover. And also, when we just look in general, reducing Tamarix cover did not reliably increase Salix cover. So for that, like, we really can't say that we can, that there is a way to compensate for it perfectly. And when we look at specific restoration methods associated with increased salix cover, we did not see a strong effect of any of those primary removal methods. Again, in cut stump sites, we saw that decrease that was associated with an increase in salix. Again, that was driven in part by sites that got worse, or sites that just kind of had changes separately from that. And overall, herbicide use was beneficial. There was an interaction with climate. But then when we're talking about environmental variables, soil properties are strong predictors of cover change for both. Overall, we really see that Salix and Tamarix just occupy different niches. Tamarix is much more of a generalist. Salix has a much narrower niche. And also, these trajectories are still incremental and ongoing, even years after removal. And, we haven't necessarily reached an equilibrium point with any of this. So just some final thoughts on all this. So in general, yes, Tamarix removal does result in a net loss of flycatcher habitat, and herbicide use is overall associated with more positive change. Again, we don't have a really good idea of how, what aspect of the herbicide use is most important, but overall it has a positive change. Removal methods on their own are not strong predictors of salix cover change in those cut stump sites, even though we did see, a proportional negative relationship. Tamarix was not fully replaced by Salix. Soil properties were very important, but overall, really, we just can't expect Salix to grow in areas where either it never grew or where conditions have since changed and it no longer can fill that niche. So some management implications of this. It is true that the biocontrol beetle does pose a danger to flycatcher habitat based on our findings. We can't count on Tamarix removal to promote Salix cover. So if the goal is just Salix regeneration and there is no flycatcher nesting in that area, then it makes sense to target Tamarix removal in cooler, wetter areas with sandier and less salty soils. And herbicide also generally seems to improve regeneration of Salix. So with that, I'd like to acknowledge, first of all, that the data that we used for this project was funded by the Marie Curie Actions Research Fellowship Program, as well as the NSF Coupled Natural Human Systems Fellowship Program. And we also want to thank a few people for helping us out with development of our project and feedback along the way. So with that, thank you for watching, and we're happy to answer any questions.